Well, hello, everyone. I'm sorry we couldn't meet today in person, but this is a great substitute under the circumstances, and these meetings are important. I've been asked by Critical Path to talk about future directions in new onset T1D. For those of you who haven't met me, my name is Joseph Hedrick, and I work at Janssen Research and Development, where I lead the type 1 diabetes effort there. I wanted to start by making the comment that we work in a T1D ecosystem. Therapies for new onset T1D are not developed in isolation by a single organization. A lot of groups have important roles, including those that you represent here today, whether it's regulators, patients, industry, or academicians. It's also important to understand that unilateral actions taken by any of these stakeholders may have an impact across this ecosystem that ultimately seeks to advance medical care and improve patients' lives. So again, that's why meetings like this, where we can at least share our thoughts, are of vital importance. So really appreciate the ability to speak here today. So I think if we want to talk about future directions, it's useful to reflect briefly on where we are today. So I think most of us would agree that T1D is primarily an autoimmune disease with metabolic consequences. The disease-causing process begins before symptoms are experienced by the affected person. And I think we'd all agree we've seen remarkable advancements and improvements in insulin delivery, and the ability to monitor insulin's effects over the last 100 years. Nonetheless, as of today, real-world control of blood glucose remains a challenge. The underlying autoimmune process is not been addressed by any therapy that's currently available to patients, and the disease remains a chronic, lifelong, progressive disease with a very high patient or caregiver burden and long-term morbidity and mortality. So if we want to look at ways that we might consider how our solutions impact patient care, one of the things we can talk about and have talked about are insulin use patterns, including dose and the stability of care. I should say that to date, as we all know, reduction in overall insulin use on its own has not been viewed by the US FDA as representing a significant clinical benefit. So one question we can consider, and we have been considering, is could measures of insulin use stability or predictability be more appropriate? So how easy is it day to day, week to week, month to month for patients to manage the disease using insulin? Are there combination endpoints that utilize insulin dose as one component, for example, the IDAC-1 score, or others that could be used as an endpoint to reflect these insulin use patterns? Whatever we use, it's important that such efforts to define these endpoints are developed in such a way that facilitate regulator evaluation. One example might be the TOMI T1D effort that's looking to develop clinical trial model simulations in new onset T1D, but is also considering potential endpoints. And then finally, I think one question that should be asked and answered is how do patients view or value reductions in insulin? If it's not important to patients, then maybe it's not important to us. I think there's been a lot of discussion and interest, particularly as continuous glucose monitors come into more common practice, how that data might be used to evaluate the impact of a therapy. Relative to HbA1c, CGM provides real-time continuous measurement of glucose control. Things that are talked about and that we can consider would be time and range, percent of time spent below a specific cutoff, or percent of time spent above a specific cutoff. However, it's important that these measures can be connected to clinical benefit and that supporting data is made available for regulatory scrutiny. I think another point is that the use of CGMs in clinical studies can be challenging. There's a large amount of data to be collected and analyzed. Blinding is an issue, especially as CGM use becomes more common practice. Patients are going to be using CGMs to manage their blood glucose. Is blinding necessary? Is it realistic? Is it even effective in such cases? And I think it's pretty clear, at least from feedback we've received, patients really don't want to be connected to two such devices if they can avoid it. I think an important consideration too is what are the differences in FDA and EMA perspectives on CGM data? Is harmonization of a CGM approach possible? And then finally, what do sponsors and clinicians need to do to move toward acceptability of CGM for clinical study and use as a potential endpoint? I think of interest to everyone, regulators have made their interest clear, is the utilization of patient-reported outcomes reflecting the patient voice. So the FDA defines patient-reported outcomes as any report of the status of a patient's health condition that comes directly from the patient without interpretation of the patient's response by a clinician or anyone else. So that definition is important. One consideration for new onset T1D, particularly as we look to pediatric new onset T1D, is collecting PRO data from children, and how does that, how do we take that into account? I think developing a validated PRO instrument for use in a clinical study that effort is not to be underestimated. It's time consuming and costly, and it may be best that it's undertaken by a consortium so that its use can be more or less standardized. I think we should consider to what extent can existing systems like PROMIS be utilized in T1D new onset studies. What's there that we can use or repurpose for what we need. And then finally, I think any PRO that's developed needs to consider the patient population in the study. For example, peds versus adults in the case of T1D, and just as important, potential cultural differences, whether that's across the U.S., between U.S. and Europe, or on a global basis, within these countries and different nations. Developing a PRO that works well across these various geographies and cultures may be challenging and needs to be considered. Along similar lines, there's been a lot of discussion about using real-world evidence, particularly in the comparison of HbA1c. So HbA1c is important. The primary advantage is that it's established that it allows assessment of a therapy's impact in the context of common practice today. The primary disadvantage is that blinded data is difficult or impossible to obtain. Obviously, this is an important tool for managing patient care. And another question is, are there potential biases introduced due to differentials in access to care? And this is an important consideration if we want to look at real-world HbA1c data. Another thing that we could think about are and again have been incorporated in many of these studies, are the frequency of hypoglycemic episodes. But how do we want to define those, and how do we want to capture those? Could we use CGM cutoffs as mentioned before? Do we want to only think about hypos that require hospitalization or outside assistance, or should we be thinking about less critical hypoglycemic events and what is the consequence of that, and again, link to outcomes for patients? Finally, any real-world evidence has to take into account the data capture, the data quality, and methods to control for confounders. These are all critical considerations when we think about using real-world evidence in clinical studies. So with those things said, where are we going? Glycated hemoglobin has been the gold standard in assessing the efficacy of diabetes clinical care. However, it provides very little insight into disease modification. And as we look at immune therapies, this is going to become critical. Some things that we might want to know: do therapies preserve residual islets or lead to restoration of islet cell mass? Are these direct effects on islets versus effects on metabolism? It may be difficult to distinguish depending on the mechanism. Have we stopped beta cell killing or revived beta cells that are dysfunctional or dormant? Can we separate these endpoints from glucose control and yet still connect them to clinical benefit? Whatever we do, decisions are going to be made based on new guidance. Can any immune therapy improve outcomes for new onset patients if the standard for comparison is glucose control in the setting of a clinical trial? Is that a realistic expectation? is this even the correct measure of efficacy for some of these therapies? What magnitude of effect will be required? What size study? For how long? The need for long-term outcomes, connection to complications. And I think this is all going to come down to will developers be willing to make the required investments. The standard of care in new onset T1D will likely change and hopefully improve as predictive or smart insulin delivery becomes more common. This could make these metabolic endpoints even more challenging to achieve in the setting of a clinical trial. Will the standard in new onset become insulin independence as a result? And finally, some of this could make intervention in the pre-symptomatic space more attractive for development as a result, something that will need to be thought about. So I don't want to end without at least bringing up some potential proposed next steps. I think identifying a forum or a mechanism to actively pursue further conversations with regulators on adequate alternative or complementary endpoints leading to drug registration is going to be important. We should work to detail areas of common ground or perceived gaps in data or proposed assessments so as to map a way through those differences. We should be able to detail reasonable, actionable items to then bring some of those areas to development and advance in a reasonable time frame, over the next 2 to 3 years, hopefully, and identify and agree on the relevant stakeholders to be involved. That really, I think, concludes the comments that I have for today. I do want to acknowledge the input of my regulatory colleague, Claudia Richard, in the content of the slides today. And again, thank you for giving me the opportunity to address the workshop today.